This tutorial is going to walk you through how to approach this question from the photosynthesis practice problems. If you haven't read it over yet, please pause the video now and take a minute to read the question, then come back to see how we're going to approach this one. This is what we call an interrupted process question. These kinds of questions are very common on the MCAT and other standardized tests because they require that you not only understand the process, but also be able to apply a logic to the analysis so you can predict what would happen if a change is made to it using a chemical or some other perturbation. So first, let's take a big picture view of photosynthesis. Light energy is used to excite the electrons in pigment molecules until a reaction center chlorophyll gets oxidized and passes its electron to an electron acceptor. In photosystem 1, the ultimate electron acceptor is NADP+, which is reduced to NADPH. In photosystem 2, the primary electron acceptor is the electron carrier plastoquinone, which is the first component in the photosynthetic electron transport chain. This photosynthetic electron transport chain transfers the electron to an oxidized chlorophyll in photosystem 1 and pumps protons across the thylakoid membrane. The proton gradient that this creates is used to power ATP synthesis via chemiosmosis, just like in the mitochondria. The energy that's stored in ATP and NADPH is then used during the Calvin cycle to reduce carbon dioxide and make carbohydrates. The enzyme Rubisco fixes a molecule of carbon dioxide to the 5-carbon molecule RUBP. The resulting molecule splits into 2 molecules of the 3-carbon compound 3PG. The energy from ATP and the energy in electrons from NADPH are used to reduce that 3PG into G3P. Some of the G3P is used to make sugars like glucose, while some is used to regenerate the RUBP. For every 6 carbon dioxides that are fixed, 1 glucose is made, and this should all balance out since glucose is a 6-carbon molecule. So overall, we have photosystem 2 passing electrons on to photosystem 1, and both of these photosystems are involved in ATP synthesis and reduction of NADP+ to NADPH. Then the Calvin cycle starts with carbon fixation to RUBP, and which produces the G3P that's used in carbohydrate synthesis. Photosystem 2 and photosystem 1 together perform non-cyclic electron transport, while photosystem 1 is responsible for cyclic electron transport. Both of these processes will generate ATP, but only non-cyclic electron transport generates NADPH. Now let's see what happens in each of the situations described in this problem. First, we're looking at a significant increase in the oxygen levels. Well, oxygen isn't required for photosynthesis to occur, so we wouldn't expect to see an increase in any of these processes. Instead, oxygen actually generates its own kind of block in this system, because it can be added to RUBP in place of carbon dioxide in the process of photorespiration. When oxygen levels go up, photorespiration goes up. When this happens, less carbon is fixed to RUBP, and in fact, you lose carbons instead of fixing them. So everything downstream in the pathway is affected, and you'll get less G3P and reduced carbohydrate synthesis too. Next, we have a perturbation that would affect a different process. In part B, we're introducing a small molecule that blocks the transfer of electrons from chlorophyll to the primary electron acceptor of photosystem 2, essentially blocking any oxidation of chlorophyll in this photosystem. Now, if we look back at our overview, we've blocked photosystem 2. So no electrons can flow through non-cyclic electron transport, and no NADPH will be made. This means that again, everything downstream would be affected. The Calvin cycle could no longer turn, so carbon fixation would also stop, and no more G3P or carbohydrates would be produced. Finally, we're asked to consider what would happen upon addition of an ionophore that can move protons across membranes down a concentration gradient. This means that the proton gradient generated by the electron transport chain could be discharged without the protons having to go through ATP synthase. So ATP synthesis would go down. Some of the protons would still go through the synthase, since it's still functional, so some ATP could be made, but it would be much less. So carbon fixed to RUBP would be converted to G3P more slowly, which means that RUBP would be regenerated more slowly, and carbohydrate synthesis would be slower. With less RUBP being made, the initial step of carbon fixation to RUBP would go down too. Since the energy isn't being captured and stored in the form of carbohydrates, it has to go somewhere else. In this case, as the protons diffuse through the membrane with the ionophore, the energy in that gradient is lost as heat. And that's that.